Good day, students. Welcome to today's lecture on the topic of lysine metabolism. Lysine is an essential amino acid that plays a crucial role in various biological processes. In this lecture, we will delve into the exploration of lysine catabolism, unraveling the genetic basis of lysosomal storage diseases associated with lysine metabolism, lysine-related epigenetic modifications, lysine metabolism in cancer, the role of lysine-derived metabolites in mitochondrial function and oxidative stress, lysine metabolism in neurodegenerative diseases, novel methods for lysine quantification and metabolic profiling, regulation of lysine transporter activity and its impact on cellular homeostasis, lysine metabolism in viral infections and antiviral therapeutic strategies, lysine bioavailability and dietary requirements, modulation of lysine metabolism and its implications for cardiac health and cardiovascular diseases, lysine and its derivatives in wound healing and tissue repair processes. Let's begin by exploring lysine catabolism. Lysine catabolism refers to the breakdown of lysine into various metabolites. The initial step involves the conversion of lysine to saccharopine by the enzyme lysine 2-oxoglutarate reductase. Saccharopine is further metabolized to alpha-aminoadipic semialdehyde through the action of aminotransferases. From alpha-aminoadipic semialdehyde, various pathways diverge, resulting in the formation of glutaryl-CoA, acetyl-CoA, or piperacolic acid. These metabolic intermediates are crucial for energy production, protein synthesis, and the synthesis of neurotransmitters. Lysosomal storage diseases, LSDs, are a group of disorders characterized by the accumulation of undegraded substances within lysosomes. Several LSDs have been associated with lysine metabolism, including cystinosis, fucosidosis, and sialic acid storage disorders. Understanding the underlying genetic abnormalities in these diseases has provided insights into lysosome function and the importance of lysine metabolism in maintaining cellular homeostasis. Lysine residues in histone proteins can undergo various post-translational modifications, including acetylation, methylation, and ubiquitination. These modifications play a crucial role in regulating gene expression and chromatin structure. For example, lysine acetylation is associated with gene activation, whereas lysine methylation can lead to gene silencing or activation, depending on the specific lysine residue and degree of methylation. Understanding the role of lysine-related epigenetic modifications provides insights into epigenetic regulation and its impact on cellular processes. Altered lysine metabolism has been observed in various types of cancer. Cancer cells exhibit increased lysine uptake and utilization to support their rapid growth and proliferation. Dysregulation of lysine metabolism enzymes and transporters contributes to the metabolic reprogramming observed in cancer. Targeting lysine metabolism may offer novel therapeutic strategies for cancer treatment. Lysine-derived metabolites, such as alpha-ketoglutarate and glutarate, play essential roles in mitochondrial function and oxidative stress response. These metabolites participate in the tricarboxylic acid cycle and contribute to ATP production and antioxidant defense mechanisms. Dysregulation of lysine metabolism and associated metabolites can disrupt mitochondrial function and increase susceptibility to oxidative stress-related diseases. Growing evidence suggests that lysine metabolism dysregulation contributes to the pathogenesis of neurodegenerative diseases, including Alzheimer's disease and Parkinson's disease. Accumulation of lysine metabolites, such as piperacolic acid, can induce neurotoxicity and impair neuronal function. Understanding the interplay between lysine metabolism and neurodegeneration may provide potential therapeutic targets for these debilitating disorders. Accurate quantification of lysine and its metabolites is essential for studying lysine metabolism. Recent advancements in analytical techniques, such as mass spectrometry-based approaches, have allowed for high throughput and comprehensive metabolic profiling. These methods enable the identification of novel lysine-derived metabolites and provide insights into their biological roles. Lysine transporters play a critical role in maintaining lysine homeostasis. Dysregulation of lysine transporter activity can lead to lysine deficiency or excess, impacting cellular processes such as protein synthesis, collagen production, and immune responses. Understanding the regulatory mechanisms governing lysine transporter activity contributes to our knowledge of cellular homeostasis and potential therapeutic interventions. Viruses exploit host lysine metabolism for their replication and survival. Lysine availability influences viral protein synthesis, viral particle formation, and immune evasion strategies. Modulating lysine metabolism or targeting lysine metabolism enzymes may offer potential antiviral therapeutic strategies. Lysine is an essential amino acid, meaning it must be obtained through the diet as the body cannot synthesize it. Adequate lysine intake is crucial for protein synthesis, collagen formation, and immune function. Understanding lysine bioavailability and dietary requirements is essential for maintaining optimal health and preventing lysine deficiency-related disorders. Emerging evidence suggests that modulating lysine metabolism can impact cardiovascular health. Dysregulation of lysine metabolism enzymes and metabolites has been implicated in cardiovascular diseases, including atherosclerosis and myocardial infarction. Targeting lysine metabolism pathways may offer new therapeutic avenues for managing cardiovascular disorders. Lysine and its derivatives, such as hydroxylysine, play crucial roles in collagen synthesis and wound healing. Lysine supplementation has been shown to promote tissue repair and improve wound healing outcomes. Understanding the mechanisms through which lysine promotes tissue repair can provide insights into developing novel therapeutic approaches for wound healing. In conclusion, lysine metabolism is a highly intricate and essential process that impacts various physiological and pathological conditions. Exploring lysine catabolism, understanding lysosomal storage diseases associated with lysine metabolism, investigating lysine-related epigenetic modifications, and studying lysine metabolism in cancer, neurodegenerative diseases, and viral infections provide valuable insights into cellular homeostasis and disease pathogenesis. Novel methods for lysine quantification, regulation of lysine transporter activity, lysine bioavailability and dietary requirements, modulation of lysine metabolism, and the role of lysine and its derivatives in wound healing further enhance our understanding of lysine's importance. Thank you for your attention, and I hope you found this lecture informative and thought-provoking.